Can somebody let me know if you can see my screen and hear me talk? I would appreciate it.
Yes.
Okay, thank you very much. If you read my announcement yesterday, for when you do the Physio X, some people have problems converting the Physio X assignment, which is the assignment you have for the cell, into a PDF file. And so I did two things. Number one, I made a short video. The videos, and I showed it to you, I think, on Monday. You know, it lasts a little over a minute. And I also made written instructions. And here are the written instructions. So when I say they're under start of course documents, that's always this first module here. Okay. You can see there's a whole bunch of stuff under here and how to upload assignments if you don't know how to upload an assignment. But these two things are there. And what happened was when I wrote the original instructions, I left off the part where you have to click on Microsoft Print to PDF and print to file. I wrote that you had to print to file, but I left this part off. And if you don't do that, then you end up with a PRN file. And in my limited computer experience, I remember we didn't have computers when I started college in '68. I don't know what a PRN file is, but that was the part I left off. And so I'll be revising those instructions. And actually, I won't have any instructions other than how to find the Physio X. And I will refer everyone to here. But once you do it a couple of times, you won't have any problems doing it. And I wanted to make sure everybody knew about that. And because I had messed up, number one, I had not published the assignments on module 2, your second module, which was chemistry. I had not published the assignments, and when I did publish them, I did not change the dates because what I do is I copy the course from one semester to the next, and I forgot to change the dates. And so if you tried to upload something, it would have said that the deadline's already passed you. And so if you read the second part of the announcement, you saw that I extended the deadline. And so the deadline is going to be extended literally for the first first couple of modules to make sure that you get those in, and there will be no penalty, but you want to go ahead and get those done. But I just wanted to show that to you and show that where it is, so that when you do start working on the Physio X in and of itself is not hard, but the converting it to from the Physio X assignment to the PDF assignment so that you can save is hard. It's a little bit hard, and if you watch the video, it's a very good video, and it'll show you an explanation. The fortunately for me and for you, this is the last big survey chapter. Okay, chapters 2 and chapters 3 assume that you know some chemistry and know some cell biology either from high school or college. Now, once we get to chapter 4 and through 16, 4 through 16, that there is no assumption. Okay, there is no assumption. We start from ground zero, sort of like we did with the first chapter. Okay, but we'll start from ground zero and go from there, and there'll be no assumption other than the terms you should that you should understand from previous chapters that you will have to have brought knowledge in there. And so I just 2 and 3 are the hardest chapters because they have so much information in them, and they're huge survey chapters. So just understand that once you get through these things, you're going to be in better shape. I had somebody from the other class, because they're going to take their first exam this week or next week, ask me if there was a exam guide. The exam guide is your student notes. The exam guide is your student notes. Those are the fill in the blank notes. That is your exam guide. 90% of the exam comes from your student notes. 90% of the exam doesn't come up verbatim. There won't be, when I said that, then I gave a test and the student thought that it was going to be a fill in the blank, and they had to know the answers of the fill in the blanks. No, that is the information you should know for the exam. With that and the slides, you have somewhere between 95 and 99% of all the stuff that's going to be on the exam. And the last 1% would be from the lectures, if I might have said anything. And I do have to say, number one, for right now, we're going to have to depend on the last semester's lectures. I'm having problems with Studio and YouTube, so I'm going to have to find me another video editor to edit the videos, but the lecture from one semester to the next is the same, so it really doesn't matter. So there is no exam guide, but there is an exam guide. That's your student notes. 90% of your exam comes from your student notes. Between the student notes and the slides, which are the slides we go over, okay? They're not the Pearson slides. I give you those just to supplement your information. And your textbook is to supplement your information. You may do your student notes, you may look at the slides, you may listen to me, and sometimes you still don't understand it. And I run across that too. And so I have to go to the textbook to supplement my information. Plus the textbook is utilized when you're doing some of your quizzes and things like that. Or sections of the textbooks utilized when you're doing some of your quizzes to give you things to read about. And so that's the hierarchy of the exam. Okay, that's the hierarchy exam. And I just wanted to explain that to you, even though we don't have the exam this week. Well, it'll probably open up next week towards the latter part of the week. Whenever it gets opened up, I'll send you a notice, and we'll talk more about the exam next week. And so we're going to start talking about this last portion, which is cell division. It shouldn't take too terribly long. There again, this makes the assumptions that you know a little bit of information, okay? They are single cell organisms and they're multi-cell organisms. And why isn't everything, why don't we just have a single cell us? Okay, why don't we have a single cell us? And it has to do with the relationship of surface area to volume. It has to do with the relationship of the surface area to volume. As we increase the volume, the surface area doesn't increase proportionally. As we increase the volume, the surface area doesn't increase proportionally. The volume increases by the cube, okay? Cube of the radius. The surface area increases by the square of the radius. And you can see as the radius gets bigger and bigger and bigger, the discrepancy gets larger and larger and larger. The problem with that is that everything has to go through the plasma membrane. Everything has to go to the plasma membrane. And so if you have something in the center of the cell, something in the center of the cell that requires oxygen, which everything requires oxygen, and has to get rid of waste, then you have a long distance for that, proportionally for that to transverse to get to the surface area. So at some point, cells outgrow their surface area, okay, and they necrose. And that's probably what happens when cancer cells, as they continue to grow, is they outgrow their surface area, okay, and outgrow their nutritional supply, okay. So that's the reason that we can't have just one single cell that's huge, and it's a relationship of surface area to volume, and one of them's a cube and one of them's a square, and you don't have to remember that. You just have to remember about the relationship surface area to volume, because the surface area doesn't increase proportionally as the volume does. Okay, and this is just an illustration of that. Cell life cycle. There are two major phases There's the interphase and there's the division phase called the mitotic phase. Okay, there's an interphase and there's a mitotic phase. And the interphase is not a phase where nothing's going on, okay? There are a lot of things going on during interphase. There are a lot of things going on during interphase. There's 3 specific cycles. But the mitotic phase is when the cell divides to form 2 identical daughter cells, So during the mitotic phase, you're going to get 2 identical daughter cells. And then if you have another mitosis, you're going to get 2 more. And so that's how we regenerate our cells, okay? With the exception of the sex cells, the sperm and the egg, which undergo meiosis, which we won't talk about in this class. We only talk about mitosis, okay? So there's 2 major phases, interphase and the mitotic phase, which is the division phase. And everybody thinks about the mitotic phase, but they don't think about interphase, okay? So interphase is a resting phase from formation of cell division, metabolically active and preparing for the next division. There are 3 stages in there. The first stage is the growth stage. Once the cell divides, you know, the nucleus reproduces, all doubles its genetic components so that can divide, and then the cell divides up its cytoplasm and its organelles. Okay, and now you have 2 smaller cells. You have 2 smaller cells. So these cells have to grow. Okay, you don't get 2 of the same size cell from a division. You get 2 smaller cells, and these cells then will grow into the size of the original cell that created the division. So the first phase in interphase, this is interphase. The first phase in interphase is carrying out the cell function and growing. Carrying out the cell function and growing. Second phase, okay, carrying out cell function and replicating DNA, okay, and replicating DNA. Until there's a need for replication, it may just stay in the growth phase where it's carrying out cell function and growing till it reaches the ideal growth, till it reaches the ideal volume to surface area ratio, and then it's just carrying out cell function, okay? But then when it reaches the S phase, the synthesis, I call it the synthesis phase. The first was G, growth phase. This is the synthesis phase. Still carrying out cell functions. Cells got to still do things. Got to do aerobic respiration. Got to have energy. Got to do all things that the cell does. The plasma membrane still selectively permeable. Okay. Got to take in nutrients. Got to get rid of waste. Okay. Now it starts replicating its genetic material. Okay. We're going to start to get ready to divide. We're going to replicate the genetic material, okay? In the G2 phase, we're still carrying out cell functions, and now we're going to synthesize our enzymes and proteins in anticipation of division. Okay, we have a G phase, an S phase, and a G2 phase. This is an interphase. During all the phases, we're carrying out cell functions. During G, the first initial G, we're growing. During the S phase, we're replicating the genetic material. And during the G2 phase, we are now synthesizing the enzymes and getting ready in anticipation of the division phase, which is called the mitotic phase. Okay. DNA replication during the S phase. Before the cell can divide, it must replicate the DNA. That copies of the DNA must be passed on to each daughter cell. We have 46 chromosomes. 46 chromosomes, we have 23 pair. We have 22 homologous pair, which means they're the same. We have two number 1 chromosomes, 2 number 2 chromosomes, 2 number 3 chromosomes, 2 number 4 chromosomes, down to chromosome number 22. The 23rd pair is the sex chromosomes, and if we're female, it may have 2 X's. If we're male, it will have an X and a Y. And these have to be replicated because our daughters, the 2 daughter cells, are the 2 progeny, are the 2 cells it's going to divide into. Because it's going to divide into 2 cells, are going to be identical copies of this. So if it's 22 homologous pairs and an XX, okay, then when it replicates, there'll be 2 daughter cells that will both be XX, okay? There won't be one that's XX and one that's XY. These are exact copy. So during mitosis is exact copy. DNA unwinds. So our DNA unwinds. Okay. Once it unwinds, we're going to make a copy of the DNA on this side and a copy of the DNA on that side. Okay. So the DNA unwinds. There's a signal for the DNA to unwind, and it unwinds. And it doesn't all unwind. Okay. It doesn't all unwind, but segments of it unwind. And then the chain starts forming, and then segments more of it unwind and chains start forming. It keeps going on like that. So the DNA unwinds, the helix untwists, and it separates the bases, okay? And we're going to pair the bases. So if we separated here and we had an A, then the new strand that's built is going to have a T. And if we had a C, so if we had adenine, we're going to have a thymine mansionate. If we had cytosine, we're going to have guanine mansionate, and so forth until we get to the end of the strand, okay? This is catalyzed by DNA polymerase. It's just an enzyme that's telling it to start. So this is going to unwind. We're going to come up with two identical strands, because this strand that unwound from here had these same base pairs, okay? And this strand that unwound from here will have these base pairs. So it's going to have two identical strands, because remember, we have to duplicate the DNA, because each one of the daughter cells, each one of the progeny, each one of the cells that result as way of the division have the exact genetic complement, okay, of the original cell. Two new strands are formed, each with one of the original strands. So this is an original strand, this was the original strand, and each with one new strand, okay? But it's an identical copy of what the original strand was. That is called semi-conservative replication, because you have one old strand and one new strand. So I might know that term in case it popped up in a true false or something, okay? Because if you had a true false and says DNA replicates by way of conservative replication, that would be false, because conservative replication would mean you'd use two original strands. Well, if you use two original strands to replicate, how would you replicate two original strands? Because you're going to have to make another new strand. And so you could never have conservative replication. I was thinking about that last night, if you could have conservative replication. Now we have two copies of all the DNA and ready for cell division of the mitotic phase or the division phase. It is in 4 stages, okay? So you can easily see that you could have a short answer question that asks you to describe the stages of interphase, and a short answer question to ask you to describe the stages of the mitotic phase. You don't have to, obviously, this is online, you don't have to do drawings, okay? But you have to be able to describe them in sentences, okay? You have to be able to describe them in sentences or bullet points, and not copy them verbatim from the notes, And we'll discuss that a little bit later. So prophase, metaphase, anaphase, telophase. Once you probably already know what they are, but once I explain them to you, you won't miss them if you happen to see a picture of them. Prophase is when the nuclear membrane is going to break down. Remember, if we're going to divide the genetic material, we've got to break the nuclear membrane down, okay? The spindle fibers are going to interact. So the centrioles, we talked about the centrioles. They were those tubule things. They had 9 tubules. There were 2 sets that were perpendicular to each other. They're going to go to the poles and arrange, and the spindles are going to be arranged. They're going to attach to the chromosomes. But in prophase, the key thing in prophase is you can start to see the genetic material. You can start to see the chromosomes. Nuclear membrane breaks down, you can start to see the chromosomes. So these are the chromosomes. They're not in any particular arrangement right now, okay? That is prophase. Prophase, the centrioles are going to migrate to the poles. The spindles are going to start being formed. Spindles are the attachments from the centrioles to the chromosomes. The nuclear membrane is going to break down, okay? And the chromosomes are going to start to become visible. That's prophase, okay? In metaphase, they line up along this line. That's called the metaphase plate. So in metaphase, the chromosomes migrate to the center of the cell and line up along the metaphase plate. In anaphase, the chromatids, okay, these are the individual chromosomes are pulled apart, okay? The centriole will be there, centriole will be there, okay? These are the spindles. The chromatids are starting to be pulled apart. So in metaphase, in prophase, we start to see them. The nuclear membrane's broken down. Spindles will start being formed. Centrioles are migrated to the poles. In metaphase, they line up along a line here. They line up along a line. In anaphase, they start being pulled apart. And then in telophase, a furrow forms, okay? Because now we've divided the nuclear material, so now we're going to divide the cell. The actual term for cell division is called cell cytokinesis. The the mitotic phase actually describes the nucleus, and technically cytokinesis describes the division of the cytoplasm in organelles. But I would say 99% of the time you see telophase talking about where the furrow forms and the cells starting to divide, and they don't use the term cytokinesis. But if you happen to see it, there it is. Cytokinesis technically talks about the division of the cytoplasm and the division of the cell organelles. So part of the mitochondria are going to go over here, part of the mitochondria are going to go over there, endoplasmic reticulum, lysosomes, and cytoplasm is going to be divided, okay? Cytokinesis, mitosis is specifically division of the nuclear material, while cytokinesis is defined as division of cytoplasm. And frequently mitosis is used to encompass both. So you may see the term telophase, and they talk about, and they don't even mention cytokinesis. They just incorporate the cell division itself into the stage of telophase. And so these are the stages. So that's all there is to the cellular division part, okay? It's not complicated. Now, if you were in a cell biology class or in genetics class, they are substages of prophase and those other things, and you'd raise prophase 1, prophase, different things, and you would learn that in more detail. But for our purposes, remember, 95% plus, up to 99% of the material on the exam comes from the student notes and the slides, okay? So for our purposes, that is what you need to learn for the exam. That is what you need to learn for the exam. Now we're going to talk about protein synthesis. We know where it occurs. We know it occurs in the rough endoplasmic reticulum, okay? Rough endoplasmic reticulum means the ribosomes attach to the endoplasmic reticulum. But we also know that the gene for the protein that needs to be made is located in the nucleus. The gene for the protein that needs to be made is located in the nucleus, but the synthesis of the protein occurs in the rough endoplasmic reticulum. So how does that occur? A little bit complicated. Proteins are made from polypeptides, multiple amino acids. The basic unit of a protein is an amino acid. Remember, it has a common structure. We went over that a couple of times. It has a central carbon with a hydrogen atom attached. It has an NH2 group, which is the amino group. It has a COOH group, which is the acid group, the carboxy acid group, and it has an R group. And the R group determines what the type of amino acid is. The R group determines the type of amino acid. An amino acid is coded by 3 of the base pairs. By 3 base pairs, it's called a triplet. So an amino acid is coded by a triplet. So the triplet CCT codes glycine, and the triplet AAA codes phenylalanine. You do not need to know that, okay? You do need to know that a triplet is a sequence of 3 nucleotides, whether it be adenine, thymine, cytosine, or guanine, okay, in whatever order is specific for that. And they have charts. That if you wanted to find out what it codes for. And sometimes there's more than one code for a specific amino acid, because there could be more than one combination of those 3 triplets, okay? More than one combination that could code for the same amino acid. And like I say, if you were in I took a course in amino acid metabolism, so you would get a chart for deciphering what the protein was. But we won't do that. But you need to know that a triplet, which is 3 of those base pairs, whether it be adenine, thymine, cytosine, or guanine, in whatever order they are, code for a specific amino acid. A gene is just a sequence of triplets that codes for a protein. So one gene codes for one protein. So in DNA, there's a gene which has a sequence of nucleotides, okay? And that sequence of nucleotides will code for a protein. If a triplet's a word, then a sequence is a sentence. A small gene is 2,000 base pairs, about 700 amino acids. So 3 in the 2,000 is about 700, okay? It's really a little bit less, but that's okay. Not all DNA codes for proteins. Some are useless introns, and some that do code for something are called exons. So we have portions of the DNA that do not code for proteins, okay? Those are called introns. So they would be a sequence that's not coding for a protein, so there wouldn't be an amino acid added in there. And so that sequence would be, it would need to be spliced out if we were gonna make a protein, okay? Or may need to be spliced out, may not need to be spliced out. It may be a signal to start or stop the protein, okay? But the exons are the ones or the triplets that do code for amino acids. We start with DNA, okay? There are 3 types of RNA. There's messenger RNA, there's ribosomal RNA, and there's transfer RNA. We need to transcribe the DNA to messenger RNA. The messenger RNA is the thing that's going to take the message from the nucleus out to the ribosome. That's messenger RNA. That's called transcription, okay? In the old days when you had stenographers, they came in with a steno pad. You took shorthand in high school or college or business school. Somebody dictated you a letter. You then went and so you transcribed their talk into shorthand, okay? So in the nucleus, the DNA, the gene for the protein that we need is going to be transcribed to messenger RNA. And then the stenographer would then take take the steno pad into their office and translate that back into what the person had said. And so that messenger RNA will take the transcribed code that's been transcribed to the messenger RNA, will take it to the ribosomal RNA in the rough endoplasmic reticulum, and then it will be translated into a protein in combination with the messenger RNA, the ribosomal RNA, and the transfer RNA. So you need to understand what transcription is and translation. Transcription is taking the code from the DNA and putting it onto an RNA messenger. Taking the code from the DNA and putting it onto an RNA messenger. The RNA messenger is going to go out of the nucleus down to the ribosome to start the protein synthesis. There's going to be a lot of terms here, and these terms are obviously useful for exams. I always have problems with my voice when I get started, especially on Wednesday. I have 3 lectures. So we need to take a closer look at transcription and translation. The transfer of information from the DNA-based sequence to the RNA-based sequence is transcription. Ribosomal RNA, there's 3 types of RNA. Ribosomal RNA, which was made in the nucleolus, that consists of 2 subunits. These are the subunits, so that's the only thing you know about it's ribosomal RNA. This is what happens here. This is the ribosomal RNA here. This is the messenger RNA that has come with the message from the nucleus, okay? This is a transfer RNA, okay? This is a transfer RNA. This is a transfer RNA right here. It has a code here. It has base pairs, and it has amino acid attached, okay? It has a code here and a base pair, and there has to be at least 20 transfer RNAs because there's 20 different amino acids, okay? And so it will come in and it will recognize this code because it will have the anti-code. This triplet's called a codon, okay? It was a triplet in the DNA. In the messenger RNA, it's called a codon, and in the transfer RNA, it's called an anti-codon. And this will bring it in here. It will park itself with the amino acid, and this is a developing protein chain with multiple amino acids here. When we talk about enzyme, this is an enzyme that's facilitating the attachment of the amino acid that's specific for this transfer RNA. And notice how is that lock and key formation, okay? So we're going to go through these specifically, but this is the big picture. This is the ribosome, this big red thing, that's the ribosome. This is the messenger RNA that has brought the complementary, okay, the complementary message from the DNA. It's being translated in the ribosome. Remember, it was transcribed in the DNA. It's transcribed in the nucleus. Now it's been translated. What does this code mean down here? What does this code mean here? It's being translated, and that translation results in transfer RNA, bringing in individual amino acids that develop a lengthening protein chain. 3 types of RNA: messenger RNA, nucleotide strand that transcribes the DNA template and something that the ribosome can translate. And we saw that. This is a schematic of a transfer RNA, okay? Cloverleaf has the amino acid attached here and has its anticodon here to read the messenger RNA codon, okay? We just continue with the same schematic. This has just got this upside down here. We transcribed in the nucleus. We brought it out to the rough endoplasmic reticulum. This is the ribosome here. We're going to translate here into the protein. Process transcription factors bind to protosilent DNA. The DNA helix is unwound for that gene, okay? It's unwound for that gene. mRNA strand begins to be synthesized. Just like DNA replication, there are specific base pairs. So for the messenger RNA, if the DNA has an A, remember RNA doesn't have a T. It doesn't have thymine. So it will have a U, okay? If the DNA has a G, the messenger RNA will have a C. If the DNA has a T, okay, messenger RNA will have an A. So you have to learn those base pairs, and you have to understand that if you're transcribing from DNA to RNA, RNA does not have thymine, okay? RNA doesn't have thymidine or thymine. Doesn't have thymine, so instead it has uracil. So everywhere there's a DNA, the message on the messenger RNA will be U, okay? It'll be uracil, because an RNA doesn't have a T, it has a U. So that's a question. That's a factoid that could come up on the exam. Okay. So here we start with the DNA template. Okay. This is the DNA template. So here's DNA. What do we have in RNA? In RNA, we have A, U, C, and G. So we have an A here, so we'd have a U over here. We have a T here, so we'd have an A over here. A G here. So G and C are the same in RNA and DNA. So we have a G here, so we'd have C there. We have C there. We have a G there. We have an A there. Remember, RNA does not have a T, so we'd have a U there. We have a T there, and we'd have an A there, okay? And so that's what our complementary RNA, this would be our messenger RNA. This would be what the code, okay, what the codon would be, okay? This is a triplet here. This is a triplet here, ATG. This codes for one amino acid. This is the codon on the messenger RNA. This is the codon on the messenger RNA. Codon, the messenger RNA sequence that corresponds to a DNA triplet. So there's a lot of terminology here. In this part, the first part, the cellular division, that wasn't hard. There's just a lot of terminology here, and you're going to have to go through it. Maybe watch a video, okay? Go to YouTube and just say DNA transcription or RNA transcription translation. Watch a 10-minute video. I do it all the time. Or go to your textbook. But it's just a matter of learning terms and maybe working through something like this, because you could very well have a problem there. You could very well have a short answer problem that says you start with a template. It wouldn't be no more complicated than 6 base pairs, okay? And then it would ask you what is the messenger RNA and what is the transfer RNA. And so you could have a short answer question. I'm not saying you will, but that's a possibility of a short answer question. So we call it a triplet for the 3 DNA base pairs. We call it a codon for the corresponding RNA base pairs, 3 of them. Okay, that's a codon. This mRNA sequence then leaves the nucleus and enters the cytoplasm. Sequence is translated from codons to the amino acids protein. Well, how does that occur? This is an example of a sheet that you'd be given if you had to translate this. You won't, this is like, this would be biochemistry. I took an elective in amino acid metabolism. That's what this sheet would be for. But this shows you an example that amino acids, okay, like this, this is alanine. It has 4 different triplets, okay, these are triplets, has 4 different triplets for what could be, okay, what could be the code for this amino acid. So, there's different triplets that can code for the same amino acid. And so we're just getting down here. In the process, there are ribosomal subunits, ribosomal RNA, okay? They attach to the mRNA and come in. We have an initiation phase, okay? tRNA has a dual recognition state. It must recognize the messenger RNA, so it has an anticodon. So if the messenger RNA has an A, a U, and a C, then the tRNA would have a U, a, and a G to be the opposite, the anticodon of the codon. And so it attaches itself to appropriate amino acid. We saw the table, the different amino acids. There's a transfer RNA for each one of those, okay? The first amino acid is alpha-methionine. Ribosomes have a room for 3 things: the incoming tRNA, the outgoing tRNA, and the messenger RNA. Elongation occurs. It then it keeps attaching to the growing chain. And then eventually it gets down to a codon and it gets down to a stop codon, okay? Once it does that, the ribosome then separates from the messenger RNA and goes back to the start of the messenger RNA and starts coding again. You can have multiple ribosomes coding on the same strand of mRNA, okay? So you can have multiple ribosomes. This is called polyribosomal complex. And this is the stages of it. It sounds a little bit complex, but if you walk yourself through it 2 or 3 times, you won't have a problem with it. Like I say, go through the notes, go through the slides, go to a YouTube video or 2 or 3, because sometimes if you listen to somebody else explain it, it makes better sense. And I'll be the first one to tell you that I do that all the time. I go to YouTube videos and have somebody else explain it, and it makes more sense to me. And so this is an example of, if you wanted to see what it looks like, these are the triplets, these are the codons, these are anticodons. But if I gave you a problem, it would only have 6. It would only have 6 base pairs, okay? I have 6 base pairs, and you would need to be able to translate what the codon is, okay, and what the anticodon is on the transfer RNA. And it would be that simple. No more than 6 base pairs. It might even just be 3 base pairs, okay? And that'd be a short answer question that you might get on the thing. And that would show your understanding of transcription and translation. That would show your understanding transcription and translation. If you could translate these base pairs, okay, which is the triplet, into a codon for the messenger RNA and into an anticodon. DNA triplet, messenger RNA codon, amino acid anticodon, DNA transcribed to messenger RNA, translated to protein. That's the big picture of the process that we just described. There's the base thing. You won't see that again except on the slides. I won't give that to you and ask you which one it translates to. That's way beyond the scope of any kind of course that we'd be teaching. I take that back. If there's biochemistry here, they might do that in biochemistry, but it's certainly not going to do this in this class. Developmental aspects of the cells. Cells differentiate. Not all DNA is used by each cell. Like I say, every cell has a complement of everything in the body. But if you're a liver cell, you use the DNA that's specific for a liver cell, but you don't use the DNA that's specific for a heart cell. Muscle cells make a lot of actin and myosin. Thyroid cells produce thyroxine. We talked about that. Or produce thyroid hormone. Cells that send out signals to each other turn genes on and off. We have atrophy. If you've ever had a broken bone, if you ever had a broken bone and you had your arm put in a cast for 6 weeks and you take it out, you'll notice you've had some atrophy of the muscle. And so that's atrophy, okay? If you have a spinal cord injury, okay, if you get atrophy of the muscles that nerve supplies. Hypertrophy is increase in size. Hyperplasia is increase in numbers and growth rate. Metaplasia is one cell type replaced by another cell type. Metaplasia is one cell type replaced by another cell type. That's usually not good. Apoptosis is a pre-programmed cell death. The cell programs itself for if it reaches a certain stage, okay, it reaches another certain stage in its life cycle, it will pre-program for death. And you can have cell necrosis if you don't have a blood supply. Cell junctions, there's 3 types of cell junctions, okay? 3 types of cell junctions. We have a tight junction, plasma membranes fuse together, and in between the fusion together of those plasma membranes, those cells, that interstitial fluid is the fluid outside the cell. Remember, we have extracellular fluid and intercellular fluid. That interstitial fluid is confined to those 2 cells, okay? And so that keeps other things out of the extracellular space. So this is a method of having an interstitial fluid between these 2 cells because of these tight junctions that allow interchange of this fluid between these 2 cells, but keep extracellular fluid from outside these cells, outside of the space here. I mean, obviously these cells are still bathed in that extracellular fluid, okay? But this one particular avenue probably for communication, okay, is sealed off to the extracellular fluid space, okay? We then have desmosomes, okay? These desmosomes are anchoring, okay? They're anchoring fibers, and what these do is keep one cell from shearing from another cell, okay? Keep one cell from shearing from another cell. And so obviously, you know, if we didn't have that, every time we rub across our skin surface, we might pull one skin surface cell from another skin surface cell, or in muscle cells, heart cells, you know, anything that's going to contract, we might have problems. So we utilize this information here, utilize this mechanism to anchor one cell to the other. They are called bolts, okay? Now for stretching and distribution of mechanical stress across many cells. And then gap junctions are tubes of straws. These are gap junctions between cells, and these are actual channels, okay, that allow exchange of nutrients, okay? So we had we had the tight junctions, which preserved the interstitial space between the cells, preserve this interstitial space between the cells. We had the desmosomes, which are bolts that prevent shearing and load off the mechanical stress. Instead of having mechanical stress on one cell, load it off onto multiple cells. And then we had the gap junctions, where actually straws or channels where you can exchange nutrients. And that's the end of that. Okay, so that is the end of chapter 3. If you weren't here ahead of time, I went over the fact that there was an announcement yesterday that the written instructions and the video instructions for converting PhysioX to a PDF file are now in the starter course documents. Starter course documents, you know where they are. They're the very first thing when you open up the modules, okay? There's a lot of useful information there. And so that's there. Because I made a mistake when I didn't publish the lab modules didn't publish the assignment parts where they could be uploaded for the lab modules for chemistry, because I made that mistake. And when I did publish them, because they were copied from the last semester, I hadn't changed the date. So even if you had done it, you couldn't upload it. I extended the deadline for the chemistry. And so the chemistry is due at the same time that the cell things are due. And for this first, this first module, this first test, I'm going to be a little bit lax about getting things in. That doesn't mean you shouldn't work on them and get them done. Okay. But the deadline for the chemistry module, for the lab modules, has been extended. Okay. So there is no late thing if you get them at the same time with the cell biology, and I'll probably even give a grace period about that. Somebody wrote me in the other class about, is there an outline for the exam? The outline for the exam is the student notes. Outline for the exam is the student notes. Okay. 90% of everything that's going to be on the exam. The exam's going to consist of, and the exam's not this week, the exam's going to consist of multiple choice, true, false, matching, and short answer. Okay, multiple choice, true, false, matching, and short answer. Usually about 2/3 of the 4 parts and about 1/3 of the short answer. Short answers are not hard. Okay, I've given you an illustration of some of the short answers in the lecture today. But that's what the exam will be like. You'll have one opportunity to take it, and we'll discuss it more next week. And you'll have 2 hours to take it once you start it, and you'll have a week to take it, a week's time frame to take it anytime you want to. It will not be proctored. The first exam won't be proctored. I don't know if I'm going to proctor any of the exams or not, but the first one is definitely not going to be proctored. So you can take it at your own convenience. So student notes. So if you haven't filled out the blanks in the student notes, then you really don't have anything to study. So come the exam. Now, questions may or may not, probably won't be lifted verbatim out of the student notes, okay? But they'll come out of the student notes. And so student notes is 90% of the test. Student notes and slides is 95 to 99% of the test. And then something that I might say in the recorded lectures, or either this semester or last semester, because I'm having problems with my editor on this semester's lectures. But the last semester's lecture is just the same. I gave the same hints on things. I'll try to get them up when I can, but you already have a recorded lecture, so you're in good shape. And most of you are attending the sessions live, which is good anyway, because you get the other stuff that I talk about. Otherwise, I don't have anything to say. Next week, we'll start on tissues. Now, the lab portion next week, we'll spend a good deal of time because we'll actually go over the tissues in the lab, like we were looking at them through the microscope. So plan on, instead of just spending the lecture time and a little bit extra, we'll probably take up most of the lab time next Monday, okay? As always, you can email me with questions. And I don't have anything else to say. Anybody have any questions for me? All right, if you don't, then I will. Okay, let me see what this says. Thank you. Okay, thank you very much too. Thank everybody for getting up at 8 o'clock. But like I say, 8 o'clock in your own house is better than getting up and fighting rush hour traffic, get to South Campus. All right, I appreciate it. Thank you very, very, very much. And I will see you next week.